smear examination. So all of this will be used by doctors enable for him to be directed towards diagnosis of certain blood disorders or hematology disorders. So ginagamit yan, no? So yung CBC, peripheral blood smear, and special lab. Now, as I have mentioned to you previously that the CBC and with the use of the peripheral blood smear, the doctor will have an idea, initial diagnosis of what is going on the patient's red blood cells, WBC, as well as the platelet. Okay? So, for example, whenever the doctor is trying to investigate anemia, gagamitin niya yung CBC, gagamitin niya rin yung peripheral blood smear. So with this different procedures or tests, it can help the doctor classify and identify what type of anemia the patient might have. Kaya pag tinanong ko sa inyo sa exam, can we use CBC to classify anemia? Yes. Can we use the peripheral blood smear to classify also anemia? Yes. Kasi sa CBC, nandun yung mga red cell indices, malalaman natin kung anong klaseng anemia meron doon sa tao. Malalaman natin kung siya yung microcytic anemia, normocytic anemia, or macrocytic type of anemia with the use of the MCV by assessing the MCV of the patient. So that is the aid or the tool that we use para ma-classify yung anemia. Diba? Now, pwede rin naman yung peripheral blood smear. Because if we evaluate the peripheral blood smear, then makikita rin natin kung mayroong mga maliliit na red cells, may mga normal na red cells, or mayroong malalaking red cells. So the sizes is also reflected, yung mga size is also reflected on the peripheral blood smear. So pwedeng gamitin yung dalawa, no? Para malaman natin ano pa anemia meron dun sa tao. Same also with the another disorder or form of RBC disorder is polycythemia. Okay? Malalaman din natin sa CBC kung mayroong polycythemia yung tao, makikita natin doon sa kanyang hematocrit, no? At saka yung mga blood counts niya. Kasi pagka polycythemic patient, you will have high counts. Okay? Mataas lahat yung count. I'm talking about polycythemia vera or the real polycythemia vera, no? One example of a polycythemic blood is polycythemia vera because there are a lot of reasons why patient might have polycythemia. Okay? Kaya meron tayong tinatawag na term na na-discuss ko na sa inyo, meron tayong tinatawag na relative polycythemia. But if we are discussing about the true polycythemia, pathologic one, we have what we call polycythemia vera, a genetic problem wherein there is an increased number of all counts, increased number of the red blood cells, WBCs, and platelet. There is a real pancytopenia in polycythemia vera. So that's why, in polycythemia vera, by the aid and the use of the CBC, pwede natin masabi na meron ng polycythemia yung tao kasi mataas lahat yung counts, no? Mataas yung WBCs, RBCs, and platelets. Is high. Mataas din yung hemoglobin, mataas din yung hematocrit. So magkakaroon ng initial diagnosis, the possibility that the patient might be suffering with polycythemia. So magagamit natin yung CBC. Yung peripheral blood smear, pwede rin bang magkaroon tayo ng, or magamit yun, at magkaroon tayo ng initial assessment that possibly the patient might have polycythemia? Yes. Because in a polycythemic individual, or if a person has polycythemia vera, then we can also see that there will be excessive number of the red blood cell. If we are trying to review blood smear of those patients, in the thin area of the smear, we see a lot of red blood cells. Okay? Minsan nag-overlap pa yung pag sobrang dami talaga. No? At makikita mo rin, if you do the estimation of the WBC as well as the platelet count using the peripheral blood smear, then we might get a very highly estimated count of the WBC to platelet. So that's why these two parameters that we use in hematology can aid us or aid the doctors in their initial diagnosis. No? Malalaman kung anong klaseng anemia or anemia ba yung tao, gamit yung CBC peripheral blood smear. Maka-classify din natin yun, gamit yung CBC at saka peripheral blood smear. Same also with the polycythemia. We can use the CBC as well as the peripheral blood smear because they are reflecting high counts. No? Kaya magagamit natin yung both CBC and the peripheral blood smear in investigation of certain disorders. Now, not only talking about polycythemia or anemia, no, because polycythemia and anemia is somehow more of an RBC disorders kasi, diba? Anemia, polycythemia. Also the CBC, as well as the peripheral blood smear, pwede rin natin ma-detect kung mayroong mga WBC problems. Like for example, if the patient is suffering with leukemia, ano naman din natin sa CBC yun, no? Ano ba ang meron sa leukemia patient, the CBC? There is a very high WBC count. So if the MedTech receive a high WBC count in an automated machine or maybe a manual WBC count, nakita niyo na nakakuha siya ng mataas na WBC count above the reference value, so possibility the person might have leukemia or maybe having an infection only. So pwede yun, no? Kaya pwedeng magamit yung CBC on that particular case. Naman natin kung yung pasyente mayroong infection o kaya may leukemia. Now, pwede rin naman natin malaman pag gamitin natin yung peripheral blood smear. So the use of the peripheral blood smear will also aid us in assessing if the patient is having infection or leukemia. As I mentioned to you last time, that whenever we are investigating problems of the WBC, such as infection or leukemia, we see immature forms of the WBC on these two particular disorders. But we have to note that if the patient is suffering with infection, the immature forms of the WBC that we see in the peripheral blood smear is not up to the blast stage. Kung leukemia, kung nakita ka ng mga immature WBCs, possibly we can see it up to the blast stage. So nakita ka ng blast, leukoblast, or maybe a monoblastic stage of WBCs. Okay? So kaya itong CBC at saka peripheral blood smear is a very powerful tool to somehow gives an initial diagnosis or assessment of what is going on the patient's blood regarding the foreign elements of the blood. Importante yung CBC at peripheral blood smear. So CBC, malaman mo kung may infection, may leukemia, no, initial diagnosis. Now, with the use also of the peripheral blood smear, pwede rin natin masabi na kung anong makita natin under the microscope, pwede masabi na maaari may infection na yung pasyente or meron ng leukemia yung pasyente. So napaka-importante na itong dalawang parameters na ito, no, yung CBC at saka peripheral blood smear in investigating diseases of the foreign elements of the blood. That is also the same with the platelets. The platelets will be, whenever you have a problem with the platelet, it can also be reflected in the CBC by determining the platelet count of the patient. Diba? So pag mababa yung platelet ng pasyente, so pwede natin masabi na baka magkaroon siya ng bleeding tendencies, especially if in a severe case of decreased platelet count. Now, it is also reflected in the peripheral blood smear. If the patient is suffering with severe thrombocytopenia, decreased platelet, so there's a possibility that makita natin sa smear, sobrang konti rin ng platelets. So kaya pala nagkakaroon ng bleeding tendency yung pasyente. So with the aid again of the CBC and peripheral blood smear, we can evaluate the entire foreign elements of the blood of our patient. What is the problem? Do you have problems with the red blood cells? Do you have problems with the WBCs? Or do you have problems with your platelet? Entirely, we can evaluate that. And the doctor uses the CBC and the peripheral blood smear in investigating or on this investigation of blood diseases, especially with regards to the foreign elements of the blood. Okay? So minsan ma-assess din natin gamit itong mga special, mga CBC at peripheral blood smear, ma-assess din natin kung nag-fail na yung bone marrow ng pasyente. No, kung ano ba yung nakikita ko. Pagka-fail na yung bone marrow, nagkakaroon na tinatawag na hypoplastic bone marrow. All counts are decreased. It will be reflected on the CBC. All counts will be decreased. So the patient might be suffering with pancytopenia. Okay? And another is, it is also reflected on the peripheral blood smear of the patient. Pag nag-estimate ka ngayon ng mga counts, like the WBC and the platelets, so mababa yung WBC and the platelet. So with the use of these two parameters, it's very helpful para ma-diagnose, para magkaroon ka ng initial interpretation of what is going on in the patient's body or in the blood of the patient. So that's why, after assessing the CBC, after assessing the peripheral blood smear, now doctor will order certain tests to confirm yung kanyang suspetsya, yung kanyang initial diagnosis. So kung for example, no, nakita doon sa kanyang CBC, nakita doon sa peripheral blood smear na merong leukemia, it is reflecting leukemia. So mag-o-order ngayon ng bone marrow sample yung doctor. So bone marrow is a special exam. So we have already discussed that. So with the aid and the assessment of the bone marrow of the patient, then mako-confirm kung meron na talagang leukemia. O kaya baka meron lang mga metastatic tumor na nandoon sa bone marrow ng pasyente. Kaya nakakita tuloy tayo ng mga immature WBC sa peripheral blood smear o sa blood ng pasyente. So hindi pala leukemia. Meron lang mga nag-i-invade ng mga cancer cells or tumor cells. Now, the term we use for that, the condition wherein the tumors are invading the bone marrow or occupying the bone marrow. Tumors, cancer cells, nag-metastasize lang, no? They are not originated in the bone marrow. They derive from other parts or other tissue, other parts of the body. Nag-invade lang ng bone marrow. So they are overpopulating now the bone marrow, occupying the space for the hematological cells, pushing the immature bone marrow cells in the peripheral blood smear. Kaya nakakita tayo ng immature forms ng mga blood cells sa blood. Dapat nasa bone marrow lang sila. So naitulak sila. So ang tawag natin doon, myeloleptic condition. So myeloleptic condition is where the bone marrow has some space occupying lesion. It may be a tumor, it may be cancer cells na nandoon na, no, sa buto ng pasyente, sa bone marrow ng isang pasyente. Kaya ngayon, yung pala, hindi pala leukemia. So kaya gagamit ng special laboratory test such as the bone marrow examination evaluation. So kaya gano'n, nag-uumpisa sa CBC at peripheral blood smear exam muna bago ngayon mag-request ng panibagong exam. So dito papasok yung mga tinatawag natin special hematological laboratory test that will support the initial diagnosis of the patient. Like for example, we have reported in our peripheral blood smear that we see, nakita tayo ng mga abnormal inclusions of the red blood cells like basophilic stipplings. So yun ang nireport natin doon sa peripheral blood smear examination natin. So maraming dahilan bakit merong basophilic stipplings, coarse basophilic stipplings in the red blood cells. Diba? Maraming dahilan doon. So maaari, baka meron siyang sideroblastic anemia. Marami kasing pwedeng dahilan bakit may basophilic stipplings. Bakit meron nun? So maaari may sideroblastic anemia, makikita rin yun, no? In certain disorders such as thalassemia. So, marami pwedeng dahilan, no? Kung bakit meron kang basophilic stipplings. So, the doctor wants to confirm what is the reason why Medtech reported that there is a basophilic stippling. So, kaya magpapa-special hematological laboratory test ka ngayon. Tingnan mo, ano ba yan? Sideroblastic anemia or thalassemia? So, pagka sideroblastic, pwede ka magpa-special stain, no? In the bone marrow, pwede rin yun, no? Magpa-collect ka ng bone marrow and then check the ion storage in the bone marrow by applying a special stain known as the Prussian blue. So yun yung mga special tests na ngayon, no? Mga special stains. Kaya yung bone marrow examination is a special exam, part of the special hematological laboratory test. Or baka naman thalassemia, no? So ngayon, mag-investigate ka, ano kayong klaseng thalassemia? Because we have different forms, different kinds of thalassemia, but the general types of thalassemia may be either alpha or beta. The most common thalassemia is the beta 1, beta thalassemia. So you know, in the beta thalassemia, there is an increased level of hemoglobin F and hemoglobin A2. So pagka ganun ang na-report, no, may mga basophilic stipplings, baka may thalassemia, beta, so magpapa-special hematology lab test ka na naman para ma-confirm mo kung meron nga talagang beta thalassemia. Tapos ni-report para sa peripheral blood smear, microcytic yung mga red cells, may mga target cells. Eh maraming pwedeng dahilan bakit microcytic yung red blood cells mo. Maraming rin dahilan bakit may target cell ka sa peripheral blood smear mo. Maraming dahilan yun. Kaya gustong i-confirm ng doktor, baka may thalassemia yung tao. So magpapa-special lab test ka na naman. That's why with the aid and with the use of this special laboratory test, mako-confirm yung diagnosis. Diba? Kaya ang una muna, CBC, peripheral blood smear, and then to confirm the initial diagnosis of the doctor, magpapa-special lab test. So we will be discussing several special lab tests sa hematology. First, discuss natin yung special exam for assessing one of the membrane defect of the red blood cell known as the paroxysmal nocturnal hemoglobinuria, PNH. We have discussed that the reason why the patient's red blood cell or why the patient has PNH because of a membrane defect in the red blood cell membrane. What is that membrane defect? Again, now that is defective in paroxysmal nocturnal hemoglobinuria. The genetic defect is known as the PIGA gene. Okay? So there is a genetic defect. Maaaring kulang yung synthesis ng gene na yun o kaya absent. That's why we have different types of PNH. Depending on the severity of the amount of this membrane protein that is not synthesized by this gene. No? Kaya meron tayong type 1, type 2, and type 3. Now, ano ba yung membrane protein na wala o maaaring kulang doon sa membrane ng red blood cell? That is the GPI protein, or to be specific, that is the GPI-linked proteins. So, if you don't have these GPI-linked proteins, then eventually, the GPI protein, which should be situated in the membrane of the red blood cell, will also be affected. Okay? Now, pwede rin sagot, anong problema ng PNH? Wala kang GPI proteins. Now, if you want to be specific, wala kang GPI-linked proteins in the RBC membrane. It may be depending on the severity of loss of these GPI proteins. Kaya meron tayong mga slight, moderate, and talagang heavy na wala, or there is an absence of the GPI proteins. Type 1, type 2, and type 3. Now, meron tayong another way of saying what is the problem with PNH. There is no certain CD markers. What are the CD markers that are affected in PNH again? So, the CD markers absent in PNH will be CD55 and CD59. So, yun ang wala, no? Ang CD55 is also known as the decaying accelerating factor, while the CD59 is the membrane inhibitor reactive lysis. Now, with the use of all of these CD markers, it makes our red blood cell protected against complement adhesion or complement attachment or complement attack. So that's why red blood cell that is absent with CD55 or 59, so the red blood cells will be very much prone to complement lysis. Kaya nga ang lalabas, magkakaroon ka ng paroxysmal nocturnal hemoglobinuria. So bakit paroxysmal? Paroxysmal means there is a transient or sometimes meron, sometimes wala, no? Kaya ang word na paroxysmal. Pagka sinabi naman natin nocturnal, so gabi, nangyayari yung hemolysis of the red blood cells, usually at night. Then hemoglobinuria is presence of hemoglobin in the urine. So those are the characteristics of this PNH. So wala kang CD55, 59, wala kang GPI-linked protein, defective yung PIGA genes. Okay? So you will have hemolysis, which now reflects that you have presence of hemoglobin in your urine samples. So again, the red blood cell are very much prone to complement attachment in certain conditions. Now, if the patient's blood may become slightly acidic and there is an activation of the complement system, so the complement may attach on the membrane of the red blood cell and then it will cause now hemolysis because of the formation of the membrane attack complex. Now, the CD55 and 59 protects our red blood cell on those particular event. Yan. Now, so what are the different tests? So meron mga confirmatory tests, meron tayong mga screening tests, no, for PNH. So the one of the screening tests for PNH is known as the sugar water test. Then the confirmatory test will be the HAM test. HAM test is also known as acidified serum test. No? Tawag din dyan acidified serum test. Now, in other textbook, the sucrose hemolysis test is somehow parehas lang ng sugar water test because the principle of these two tests ay almost the same. Kaya sa ibang libro, sinasabi nila na sugar water test or sucrose hemolysis test can be a screening test for PNH. But in the Barbara Brown textbook for hematology, the sucrose hemolysis test is a confirmatory test. But if you will be choosing between HAMS test and sucrose hemolysis test, which is the best answer as a confirmatory test for PNH? So your answer will be HAMS test or acidified serum test. Iwanan ba? Now, there is another confirmatory test for PNH, and this is by the use of the flow cytometrical analysis. Because in the flow cytometry, the principle of that is detecting the presence of the CD markers of our blood cells. Okay, mga CD markers. So pagka walang CD marker na 55 o kaya 59, so possibility the person might have PNH. Kasi ang CD55 at 59 hindi lang nakikita sa red blood cell membrane. Pag meron kang PNH, wala rin yung mga WBCs. Nawawala rin yung CD55 at 59. So yun ang isa sa ginagamit to confirm if the patient is suffering with PNH with the use of flow cytometry, detecting the presence or absence of CD59 or 55. Nakintindihan ba? So confirmatory test din yun. Kaya pag tinanong sa inyo, what are the confirmatory tests for PNH? Pwede yung HAMS test, pwede yung flow cytometry, detecting the presence of CD55 or 59. Pwede rin ba yung sucrose hemolysis test? Pwede na rin, kasi sabi ni Barbara Brown yun, na sucrose hemolysis test will be part of the confirmatory test. Pero the best answer, HAMS test and flow cytometry for confirming PNH. Okay, now, so what is the principle of the sugar water test? And what is the endpoint of the sugar water test? The same also with the sucrose hemolysis test. What is the principle of the sucrose hemolysis and what is the endpoint of the sucrose hemolysis test? So the principle of the sugar water and sucrose hemolysis test. So we will be, from the word itself, we have what we call the sugar water, okay, and sucrose. So we will be preparing a sugar solution. Okay, so water plus sugar, table sugar, kung wala nun, so sucrose. Now, the red blood cell, whenever they are subjected into a sugar environment, they are very much prone to complement attachment. Okay? So mag-a-attach yung complement, then the complement will be activated and causing now destruction of the red blood cell. So that's why the endpoint of sugar water and sucrose hemolysis test will be hemolysis. Okay? So ang positive result ng sugar water and sucrose hemolysis will be hemolysis. So that's why sensitive ito kasi, itong dalawang test na to, sensitive yan sa PNH. Kaya pag nag-positive sa sugar water test, you have to confirm it with HAMS test because sugar water test, as a screening test, pwede rin mag-positive yung ibang mga hematological disorders. Okay, pwede mag-positive yung ibang myeloproliferative diseases. Pag sinabi natin myeloproliferative diseases, mga examples ng mga leukemia, no? So pwede mag-positive. So pwede rin mag-positive yung ibang, like for example, Hodgkin's lymphoma. Pwede rin mag-positive sa sugar water test. That's why it is not specific for PNH, but it is sensitive for PNH. So the patient is suffering with that disorder, magpa-positive yan sa sugar water test. That's why we have to confirm it using now the HAMS test or HAM test. Sa ibang libro kasi HAMS test. HAM test or HAMS test or acidified serum test. So pag nag-negative sa sugar water test or sucrose hemolysis test, so no need for a confirmatory test because PNH is highly sensitive with sugar water. So pag negative, no need for confirmation. So pag negative, wala siyang PNH. So iba siguro ang dahilan kung bakit ka nagkakaroon ng hemoglobinuria or hemolysis in your red blood cell. Diyan ba? Yan. So yan ang principle lang ng PNH. So may sugar solution, so you place the red blood cell of the patient suspected with PNH. And then yung plasma niya, may mga ano yun may mga complement or serum niya, serum pala, no, serum. Okay? The serum. So ang mixture nito, sugar water, red blood cell of the patient, and serum. The serum is the source of the complement, and then the endpoint of that will be hemolysis. Okay, so serum ang ginagamit natin dito. Now, so again, pag nag-positive sa sugar water test, so now go with the HAMS test or acidified serum test. Now, so what's the principle of this test? So gagamit tayo na, I will just merely simplify this, no? Yung principle ng acidified. So sa acidified serum test, gagamit tayo ng tatlong different kinds of serum. We will be using the patient serum, normal serum, and then we have the inactivated serum of inactivated serum. Now, how do we prepare first the inactivated serum? So inactivated serum is prepared by heating the serum at 56 degrees Celsius for 30 minutes. Okay, 56 degrees Celsius for 30 minutes. Now, the objective of that is to inactivate the complement proteins. So mawawala yung function, no? O masisira yung mga other complement proteins that is in the serum of the patient. Diba? Yan. So yan ang ano natin. So sa patient serum, nandun pa yung complement, no? Sa normal serum, coming from other individual na normal, nandun pa rin yung complement. Pero sa inactivated serum, wala nang complement activity dyan kasi sinira natin by heat treatment. So maraming paraan para ma-inactivate yung serum. One of that is heat treatment. Heating it at 56 degrees Celsius for 30 minutes. Deactivating the different complement proteins. Okay, na-prepare mo na, no? Now, then we use now the fibrinated blood. Okay, so gagamit tayo ng blood ngayon. You will be putting not blood pala. You have to acidify it first. Kaya nga acidified serum test. So you will be putting around 0.2 normal, okay, of HCl. Okay, you will be putting it there, no, HCl. And then ma-acidify na siya. Kaya nga acidified serum test. Then after that, place now around 20 UL of the patient's red cells. Okay, so 20 UL of the blood. Then incubate this. Maybe incubate this at 37 degrees Celsius or maybe at a room temperature. Diba? So incubate them. Then wait for hemolysis. So check kung saan nangyari yung hemolysis. So patient serum, normal serum, and inactivated serum. Now, so if the result is like this, positive hemolysis on patient serum, positive hemolysis on normal serum, but there is no hemolysis in inactivated serum after incubation, then this is paroxysmal nocturnal hemoglobinuria. So that's why the expected result in if the patient has PNH, there is no hemolysis on inactivated serum, but there will be hemolysis on other serums because those serums, like patient serum and normal serum, still containing complement proteins. Yan ba? Yan. Kaya ang result ng PNH. Kaya pagka sinabi sa inyo, what is true about PNH? PNH is complement-dependent hemolysis. It is a complement-dependent, but not complement-independent. Okay? So, now, but there are also other disorders that can also give us a positive result for HAMS test, like CDA type 2 or hereditary spherocytosis. But they give us different kinds of result. Pero nagpa-positive sila sa acidified serum. Okay? Yan. So the same thing, no? Lagyan mo yung prepare serum and then add acid and then add the red blood cell. Now, but if the result is like this, there is no hemolysis on all patient serum, but there is hemolysis on the other forms of serum, then this is CDA type 2. Okay? There are 3 different types of CDA, type 1, type 2, and type 3. CDA is also known as the congenital dyserythropoietic anemia. Okay, congenital disk erythropoietic anemia. The problem with CDA is the development of the red blood cell. It is a genetic problem wherein the nucleus of a maturing red blood cell is impaired. There's an abnormal nuclear development. An abnormal nuclear development. It may exhibit binucleated or multinucleated red cells. Remember that a normal red blood cell na nasa bone marrow natin, whenever they are immature, they must contain only one nucleus. But in several types or certain types of the CDA, the nucleus may be binucleus. Maaring dalawa yung nucleus dun sa isang red blood cell, na immature red cell. Dapat isa lang yung nucleus. Dalawa, o kaya multinucleated, maraming nucleus sa isang immature red cell. It will turn out to be abnormal. Magkakos ng anemia yun. Yan ba? Now, so we have three types, no? Type 1, type 2, type 3. But type 2 is the only one that gives positive result for acidified serum. That's why CDA type 2 is also known as HEMPAS. Okay? So tawag din sa kanya ay HEMPAS. What is the meaning of HEMPAS? HEMPAS is also known as hereditary erythroblast with multinuclearity and positive acidified serum. Okay? So that is the meaning of HEMPAS. So between the three different types of CDA, sino ang HEMPAS doon or ano yung tinatawag na HEMPAS? So that will be type 2 because it caused positive acidified serum test. No, it gives a positive. So what is again a positive result? Hemolysis, but it gives different hemolytic pattern compared to paroxysmal nocturnal. So in a paroxysmal nocturnal, no hemolysis on inactivated serum. Now, how about in hereditary spherocytosis? So in hereditary spherocytosis, it will also give us a positive result in acidified serum. As you can see here in our illustration, it may exhibit hemolysis on different forms of serum. So magpa-positive naman siya sa lahat. So pag gano'n ng report, pag gano'n ng result, so baka hereditary spherocytosis yun. But hereditary spherocytosis is, but Ham's test or acidified serum test is not a confirmatory test to diagnose or a test to diagnose hereditary spherocyte. There is another test, special exam for hereditary spherocytosis. Nagkataon lang na pag meron kang hereditary spherocytosis, nagpa-positive din sa acidified serum. It shows hemolytic pattern or hemolysis on this test, but it shows different pattern of hemolysis if you compare it with PNH. Yan ba? So yan ang principle ng acidified serum test, which is used as a confirmatory test for PNH. So let us now go with hereditary spherocytosis. So test for hereditary spherocyte. Now, hereditary spherocyte is also an abnormal membrane defect. It is also a genetic problem causing deficiency or abnormal molecular structure of membrane proteins. What are the membrane proteins that might be affected in hereditary spherocytosis? One of that is spectrin. We have two forms or two chains of spectrin located in the peripheral portion of our RBC membrane, alpha and beta. So it depends on the molecular abnormality. Maybe the defective is the alpha chain or maybe the beta chain or both of that affected yung spectrin. That's what generally, hereditary spherocytosis, there is a molecular defect or maybe there is a deficiency of either alpha or beta chain. So yun ang problema sa hereditary spherocytes. But there are also other membrane proteins attached to the spectrin, which is, if these membrane proteins are defective, then it will also affect the integrity of the spectrin proteins. So what are the other proteins that may also lead to hereditary spherocytosis? So one of that is protein band 3, okay? Another is protein band 4.2, and another is ankylin. So magkakadikit kasi itong mga proteins na ito, yung spectrin, protein band 3, protein band 4.2, and ankylin. So that's why if you have defects on all of this protein, then it may lead to instability of the RBC membrane. It will now cause an abnormal poikilocyte known as spherocytic red blood cell. That's why, if I ask you what are the different proteins, if defective, may lead to spherocytic formation of the red blood cell. So you may answer spectrin, ankylin, protein band 3, and protein band 4.2. But the best answer among all of these membrane proteins will be spectrin. Now what happens in the red blood cell whenever you have these defective membrane proteins? So the red blood cell's membrane becomes loose. As it travels in the circulation, the red blood cells membrane, because it is loose, so nagkakaroon ng blebs. Okay? So like this. So the red blood cell may form blebs. yung mga yan. And okay, mga protrusion, no? As they travel along the circulation. Okay? Now, eventually, if they go inside the spleen, okay, sa spleen, so discuss natin na yung environment ng spleen is not really conducive for the red blood cells. That's why in able for the red blood cell to survive in the harsh environment within the spleen, no, harsh environment. Kasi sa spleen, mababa yung glucose sa tissue niya, no? Medyo mababa yung glucose level. Then medyo mababa yung oxygen tension. Okay? And then maliliit yung mga capillaries or the ang tawag na splenic sieve. Maliliit yung mga blood vessels there, no? Okay? And there's a little bit there's an acidic environment also on the spleen. So these are not conducive for RBC survival if your RBC is senescent. Medyo matanda na yung red blood cell, no? Malapit na ma-expire. At hindi rin conducive yung environment ng spleen if you have other problems with your red blood cells. Pero pag mga young pa yung mga red cells, no, hindi pa malapit ma-expire yung mga red blood cells within our circulation kasi may lifespan yan, so nakaka-survive in the spleen. Now, in the spleen, also, there are macrophages na nag-guard, no, doon sa circulation. Kaya nakikita nila pag may abnormality, yung ating mga red blood cells, may mga abnormal inclusions, may malalaking inclusions. So the macrophages within the splenic tissue can have this opportunity to check the red cells within that splenic sieve because the blood flow on that area, dahil sa maliliit nga yung mga blood vessels there, medyo bumabagal yung flow ng blood doon. So merong chance yung mga macrophage na matingnan yung mga RBC. So magkakaroon sila ng contact doon sa mga red blood cells. Now, one of the problems in hereditary spherocytosis, because the membrane are loose due to the defects of these peripheral proteins, such as spectrin proteins, so the RBC membrane becomes loose, forming now blebs or protrusion on the membrane, no? So, pag dumating sa spleen, itong mga red blood cells na to, so possibility, the macrophages will remove those blebs. Okay? So, kasi na-expose yung tinatawag nating mga possibility of the eat-me signals. No, pwedeng ma-expose yan. Like, for example, because of the loosening of the membrane, exposing yung phosphatidyl-serine. So, malalapit yung macrophage. So, kakainin niya yung blebs, no? Possibility eating the blebs ng ating mga red blood cells. So, pag kinain yung blebs, o yung mga protrusion na yan, pag kinain ng macrophages, yung membrane kasi ng red blood cells natin, they can, parang after makain, nag-ano sila na fuse back. So, merong capability yung ating RBC membrane that they can fuse back. Now, but fragments or part of the membrane is lost. Hindi ba? Part of the red cell membrane is lost. Kaya nakakain unti-unti yung membrane ng red cells. Kaya ano mangyayari sa red blood cell? So, kanina may bleb siya tinanggal. So, pwedeng paliit yung ating mga red blood cells. Kasi unti-unti nakakain yung membrane. Kaya what will happen to our red blood cell? They become small. No? So, liliit sila ngayon. Okay? So, they will become small because loosening of the membrane. That's why, if the red blood cell now becomes small, the surface area, okay, the surface area to volume ratio, okay, the surface area to volume ratio of a spherocytic red blood cell will become what? So it will be decreased. So ibig sabihin nun, lumiit yung red cell, yung laman sa loob ng RBC cannot anymore contain, or there's no enough space for the volume or for the hemoglobin there within that red cells. No, hindi na ma-contain kasi lumiit yung red cells. So, palit yung red cells, hindi na ma-contain yung laman sa loob. That's why a spherocytic RBC loses its central pallor. Kaya pagka in-evaluate natin ang peripheral blood smear, nakakita kayo ng spherocyte. Ang blood picture reflected will be microcytic hyperchromic. Microcytic, why? Because the red cell is very small and there is no more central pallor. So that is possibility that the blood picture reflected will be microcytic hyperchromic red cells. That's why if you will now determine the MCV as well as the MCHC of hereditary spherocytes or spherocytic red cells, MCV is low, MCHC will be high. Now, what will be the consequence if your red cell is like this? So yung membrane niya magiging mas fragile. Okay, so there is an increased fragility of the membrane of the red blood cells. That's why in spherocytic RBC, there will be increased fragility. Madaling pumutok na ang red blood cells na ito. No? Kumbaga, sabi nga sa ibang libro, it is a bag full of content inside. It is a small bag na masyado ng maraming laman sa loob. Okay? Or maybe a balloon, no? Na yung balloon, diba, nag-stretch siya. Pagka sumobra na dami ng air inside the balloon, it will eventually burst. So that is what is going on, no? With the spirochetic RBC. As the membrane loses its, or as the red blood cell membrane lose or decrease in area, so nagiging fragile. Pag itong spirochetic RBC na to, tumama ngayon sa mga blood vessel wall, pagpunta niya sa circulation, sa heart, yung valve doon, causes turbulence, pwede sila tumama doon sa mga valves, walls of the heart, walls of the blood vessels. There's a possibility that if they bump on the blood vessel wall, in heart valves, in heart walls, possibility they will lapse. That's why patient may suffer with hemolytic anemia. So magkakaroon ng hemolysis. So yan ang pathophysiology ng spirochetic RBC. It can cause hemolysis. Alam ba? Hindi intravascular hemolysis pagka nasira within the circulatory system. Now, because these RBCs are so small, they cannot contain inside anymore within itself the volume or the amount of hemoglobin there. So if this spirochetic RBC will be subjected to a hypotonic solution, so you know the travel of fluid in a hypotonic solution, right? So the fluid will go inside the red cell and it can cause now hemolysis to a cell. So papasok ngayon ang tubig doon sa RBC. Tapos, puputok siya. Kaya mabilis pumutok pagka pumasok na yung fluid. That is what happened in the fluid. Diba? Pagka hypertonic yung solution, ang mangyayari, magsishrink yung red cells. Pero pagka hypotonic yung solution, nilagay natin yung red blood cell na hypotonic solution, what will happen is that the fluid will go inside the red cell and then it can cause now swelling. Now, a normal red cell can withstand meron lang volume of fluid that can hold by a normal red cells. Our red cells can stretch. Pero pagka severe na yung hypotonic solution or hypotonicity of the solution, mas maraming fluid na papasok, meron lang time na mag-burst na siya. So a normal red cell can still withstand or can hold amount of fluid in certain point. But in spirochetic RBC, konting fluid pa lang pumasok sa kanya, may possibility pumutok na siya. So yan ang principle ng tinatawag natin EOFT studies or erythrocyte osmotic fragility test. So we are trying to determine if the membrane of the red cell is fragile or not. So expected in a spirochetic RBC, the membrane are so fragile that if you subject it into a hypotonic solution, it will easily burst. So what is the principle of the EOFT? So in EOFT studies, we will be using heparinized blood. Why is it heparin is the ideal anticoagulant for this test? Because heparin prevents the change in the pH of the blood. Because alteration in the pH of the blood may happen if you use other anticoagulants. No? And whenever there is a change in the pH of the blood, so there's a possibility of changing the shape or appearance of the red blood cell. It may cause a little shrinkage of the red blood cell. Okay? So that's why we have to use heparin. Not enable for it not to alter the pH of the blood. Enable for it to maintain the appearance of the red cells. Okay? Kasi para hindi lumaki o lumiit yung red cells, importante ma-maintain kung ano yung talagang nandoon sa patient sample. Ano ba ang appearance or shape ng red cell doon sa patient sample suspected with hereditary spherocytosis. Okay? So that's why the blood should be heparinized and it should be defibrinated. What is the meaning of defibrination? Defibrination is removal of the fibrin clot. So kailangan yun ang kompletong characteristic of the blood specimen that we have to use in EOFT, heparinized defibrinated blood. Now, so you have to prepare that. And what else will you prepare? Using a 12 by 75 millimeter tube, glass tube. Okay? 12 by 75 millimeter glass tube. So yun dapat ang gagamitin natin, 12 by 75 millimeter glass tube. So dapat yun ang size na, no? Mag-prepare tayo ng different salt concentration. Okay? Different salt concentration. From 1, pwedeng mag-start ka sa 1, no? That is a hypertonic solution, no? 1% sodium chloride. Okay? Or maybe you can just start on a hypotonic solution such as 0.95 or 0.85%. Okay? So pwede yun, no? So from hypertonic pa isotonic yung solution mo. Kaya from 1, now from 1%, okay, up to 0% of sodium chloride. So the reagent that we use here will be a sodium chloride. Okay? And so sodium chloride salt solution ang gagamitin natin. Now, So, ang gagawin mo, 0.1. So, prepare ka, 0.1, 0.95, 0.9, 0.85, 0.8, 0.75, etc. hanggang sa maging 0%. So, you can use that. Or maybe you start, as I mentioned to you, sa isotonic na, no? 0.9 or 0.85. Kasi we are expecting that a normal red cell should not lies in an isotonic fluid. Okay? So after you have prepared the different concentration of salt, then put now around 20 UL of heparinized defibrinated blood on all of the tubes. So ito yung principle ng EOFT, no? So prepare a different concentration from hypertonic or maybe from isotonic to a hypotonic solution, and then you put 20 UL of the blood on the different test tubes. Then incubate for around 2 hours. So medyo matagal to, no? So pwedeng 1 hour to 2 hours incubation, and then the endpoint of that is hemolysis. So we will now check the hemolytic pattern or hemolysis, what tube shows or what concentration of salt nagkaroon ng hemolysis. We have a normal reference value. A normal red cell will start lysis. Magsisimulang malyse ang mga red cells. Tawag natin doon initial or partial hemolysis is expected. We see that if the concentration of salt is around 0.40 to 0.45%, yun ang normal. So ibig sabihin, ang normal na red blood cell, pag nilagay natin sa 0.5, 0.6, 0.7 na concentration of salt, it can hold water and it will not cause hemolysis. But it will now only show hemolysis at 0.40 to 0.45%. And we call it partial hemolysis. Now, what is the meaning of partial hemolysis? Partial hemolysis is the first test tube from, so ang pagbabasa, no? Ang pagbabasa natin, okay? So we are expecting that there will be no hemolysis on a hypertonic solution if you have prepared a hypertonic one, because the red cell there will shrink. Lalabas kasi yung fluid. So magsishrink dapat. So wala dapat na hemolysis doon, no? So ang pagbabasa natin, check from hypertonic, check mo saan ba nag-show ng hemolysis, partial hemolysis. So from hypertonic pa isotonic ang pagbabasa. Hindi ba? So kunwari, nakita mo, meron ng hemolysis doon sa 0.40 or 0.45%. Partial hemolysis, that is the first test tube, no, that shows hemolysis. So what is partial hemolysis? There is a redness in the solution, but not all of the RBCs are lyse. So there are still red cell button. So mayroong mga RBC pa na hindi na lyse na nasa ilalim ng test tube. So titignan mo, sino yung first test tube na may ganun? Okay? So makikita natin dapat yun, yung partial hemolysis at 0.40 to 0.45% sodium chloride. Okay? And then after that, record it. Then after that, tignan mo ulit, papunta ka ulit sa hypotonic ka ulit. Hahanapin mo yung first test tube naman na nag-cause ng complete hemolysis. What is the meaning of complete hemolysis? Complete hemolysis is red yung fluid at wala ng red cell button. Wala ng red cell sa ilalim nung RBC. Ay, nung test tube pala. Okay? So, ibig sabihin, lahat ng RBC na lyse at mangyayari dapat yung complete hemolysis at 0.30 to 0.35% sodium chloride. So that's why the reference value should be showing us, okay, if the RBC is normal, there is no spirocytic RBC, there is no hereditary spirocytosis, so it should show us a partial hemolysis at 0.40 to 0.45%, and complete hemolysis should happen at 0.30 to 0.35% sodium chloride concentration. Dapat mangyari yun. Now, in hereditary spirocytosis, early yung kanyang hemolysis. Baka makakita tayo ng hemolysis sa 0.5 pa lang. Meron ng initial hemolysis. Okay? So pag ganun ang binigay ko sa inyo sa exam, for example, the result of EOFT shows that there is already hemolysis on 0.5% sodium chloride o kaya 0.6% sodium chloride. May hemolysis na kagad. Remember that our red blood cell can withstand no hemolysis at 0.5% sodium chloride. Because if you subject a normal red cell in a 0.5%, a hypotonic nun. So the fluid will go inside the red cells. Diba? Pero the red blood cell, remember, it can stretch. That's why it can still hold water at 0.5% sodium chloride. Pero pagdating na sa 0.40 to 0.45, papasok pa lalo yung mga water, so magla-lyse na siya. So ang normal, 0.5, wala muna dapat. Pero pag nakita natin na yung mga RBCs na hihimulize na earlier, 0.5, 0.6%, nung may hemolysis na, nakikita, may initial hemolysis. So what is again initial hemolysis? The fluid is already showing as red tinge, but there is still a red blood cell at the bottom of the tube. So that is what we call initial hemolysis. But it is early, 0.5, 0.6, nakita mo na. So maaaring increase ang fragility ng RBC mo. And we see that in spirocytic RBCs. So may mga spirocytes ka. And it is possibly, baka merong hereditary spirocytes yung tao. Intindihan ba? Yan. So that is the principle of the EOFT. We are subjecting the red blood cell at different concentration of salt, and then we will try to check saan nangyaring partial hemolysis or initial hemolysis, saan nangyaring concentration of salt yung complete hemolysis. Diyan ba? So, ganun ang principle ng EOFT, no? Now, that's why, in an EOFT study, if the patient is suffering with hereditary spherocyte, the expected result will be increased sensitivity in EOFT. Pag sinabi natin increased sensitivity in the EOFT, it means fragile yung red blood cells. Early yung hemolytic pattern na nangyari. Tatawag doon, increased sensitivity. Okay? The hereditary spherocyte has increased sensitivity to EOFT studies. Now, another way of saying, hereditary spherocyte has increased fragility and decreased resistance. Okay? So decrease naman yung resistance niya kasi madali siyang pumutok. Unlike with the normal red cells, it has a good resistance in hemolysis kasi it can still hold water at 0.5% sodium chloride. O ba yun? And now, there are other poikilocytes aside from spherocyte that shares the same result with EOFT, meaning they also shows increased sensitivity or increased fragility or decreased resistance to an EOFT study. What are the other poikilocytes? Pag tinanong ko sa inyo sa quiz, what are the other poikilocytes or sa exam? So your answer will be what? Macrocytes, ovarocytes, and elliptocytes. So if the patient's blood do also have these different forms of poikilocytes, then possibility you will expect to have an increased sensitivity or reading in the EOFT, meaning they also give us increased fragility and decreased resistance of their RBC membrane in EOFT studies. Yeah, pero pwede rin mag-positive sa EOFT or increase ang reading ng merong macrocytes, merong macroovalocytes o kaya merong elliptocytes. That's why patients suffering with megaloblastic anemia, because in megaloblastic anemia, we see macroovalocytes in the peripheral blood smear of the patient or in the blood specimen. So if you subject this, the blood of the patient that suffers with megaloblastic anemia in EOFT studies, it will also give an increased EOFT result or report, okay, or increased sensitivity to EOFT. The same also if the patient do have hereditary elliptocytosis. Okay, hereditary elliptocytes will also give us an increased sensitivity or reading in the EOFT studies. But megaloblastic anemia, okay, is not confirmed by EOFT studies. So EOFT is used to confirm hereditary or diagnose hereditary spherocytosis. Iba ang ginagamit natin test sa megaloblastic anemia to assess megaloblastic or confirming megaloblastic anemia. Natiyempo lang na pag may mga macrocytes, possible na mag-positive sa or increase ng sensitivity sa EOFT studies. Pag may mga macrocytes. So isang example lang may mga macrocytic RBC or may mga macroovalocytes ay yung megaloblastic anemia. Naintindihan ba? Okay. Now, meron din naman mga other poikilocyte that shows naman, no, decreased sensitivity to EOFT studies or increase ang kanilang resistance, decrease ang fragility. What are these poikilocytes? No, what are these poikilocytes that has that characteristic? These are collectively called as non-lysing RBCs. What are the examples of non-lysing RBCs? I've already mentioned that in our lectures. Okay, what are examples of non-lysing RBCs? We have target cells, sickle cells, severely hypochromic red cells. So if the patient's blood, no, merong mga ganitong klase ng abnormality in the RBCs, kaya ano yun, may mga target cells, merong severely hypochromic red cells, may mga sickle cells. These RBCs, pag pumasok sa kanila yung fluid, if you subject them into a hypotonic solution, they have a lot of spaces inside them. That's why they can hold more water than the normal red blood cells. That's why what we see sometimes on those patients having target cells, numerous target cells on their blood, okay, what we see if you subject it into an EOFT studies, we see that the initial hemolysis might happen later. So dapat ang normal, initial hemolysis or partial hemolysis mangyari sa 0.40 to 0.45. So maaari ba yung initial hemolysis mangyari sa 0.30, 0.35? Initial hemolysis. Dapat complete hemolysis na yun. Diyan ba? So kaya decrease ang reading or sensitivity ng sa EOFT ng mga poikilocyte na ganito. No, pag meron ka mga non-lysing RBCs. Now, saan ba nakikita ang target cells? Usually we see target cells on certain disorders such as we see that in sa mga thalassemia. Kapag meron ka mga minor thalassemia, pwedeng makakita ng target cell. Kapag may mga minor thalassemia ka, baka pwede ang makikita nating result sa EOFT kung na-subject sila sa EOFT studies, so you will have a decreased reading or sensitivity to EOFT studies because their membrane is said to have an increased resistance and decreased fragility if you subject it again on the EOFT studies. Okay? So maraming sakit na makikitaan natin ng target cell, no? Isa dyan yung thalassemia, no? Like for example, minor thalassemia. Then, How about yung mga anemia na may hypochromic? As I mentioned to you, severely hypochromic red cells can also give us a decreased sensitivity to EFT studies. So that's why those anemias that showing us that show us hypochromic red cells may give us a decreased reading of the EOFT studies or decreased sensitivity in the EOFT. Again, what are those anemia that has hypochromic red cells? One is iron deficiency, one is sideroblastic anemia, another is thalassemia, no? And then another is anemia of chronic disease or anemia of chronic inflammation. So that's why if the disease of the patient is this 4, giving us a hypochromic red cells, then you subject it to an EOFT. Possibly you might have a decreased EOFT result or reading or sensitivity to the EOFT reading. Naintindihan ba? Pero yung mga hypochromic anemias na yon, hindi ginagamit ang EOFT to diagnose those disorder, yung mga hypochromic anemia. There are other tests that will be used to confirm these hypochromic anemias. So if I ask you what is the clinical use of the EOFT, the clinical use of the EOFT or significance of the EOFT is to diagnose hereditary spherocytosis. Diyan ba? So yan ang principle ng EOFT. Now, so in the laboratory, you can prepare, no? So meron na minsan na nakaprepare ng 1%, 0.95, so meron na mga different concentration of salt. Or maybe you can do this, okay? Meron na kasi mga ganyan nakaprepare na eh. Or maybe, you place ganito, no? Pero kasi yung principle na gamit ka ng test tubes. Okay? So you have test tubes, no? Number it from 25. Okay, test tube number 25, 24, 23, 22, okay, 21, no? Hanggang 12, number 12. Okay, so 20, 19, 18, 17, 16, 15, 13, 14, 13, 12. Ulitin ko, no? So you will be preparing test tubes from number 25 to 12. So number your test tube from number 25 to 12. So you have 25, 24, 23, okay, 22, 21, then 20, 19, 18, 17, 16, 15, 14, simula ko na lahat, 13, and then 12. Okay? So prepare ka ng test tube. So bawat isa test tube yan. Test tube, number mo, no? Hanggang sa 12. Okay? Now, so the number of the test tube is the total number of drops that you put on that test tube. Okay? So kung 25, ibig sabihin, may 25 drops. Ang lalagay mo muna dito ay yung, so ang reagent na gagamitin natin, 0.5% sodium chloride. Okay? So the reagent we use here, this is a manual method. Kasi sabi ko sa inyo kanina, meron na sa lab na nakaprepare na yung different concentration of salt that you will be using for EOFT studies. May 1% na, 0.85, so nakaprepare na, no? Pero kunwari, wala kayong ganun na nakaprepare ng mga salt concentration na sa lab for EOFT studies. You only have 0.5%. Okay? So 0.5% lang. So the initial concentration that you have is 0.5% sodium chloride. So the number of the test tube here will be indicating how many drops of 0.5% sodium chloride you will use. Okay, so for example, if it is 25, so put 25 drops of that 0.5% sodium chloride. So next test tube, number 24, 24 drops. Okay, tapos, pag 23, 23 drops. 22, 22 drops. So 21 and so on and so forth. So sa 12 test tube, number 12, no? Yun ang nakalagay sa label. Number 12 test tube, put 12 drops of the 0.5%. Now, the total number of drops should be around, dapat ang total number niya nasa mga 25, no? So kaya ang total number dito, 25 sa 0.5%. Okay, now, so kung 12 dito, ilang drops ng blood ang ilalagay mo? Okay? So, ilang drops ng blood? So, ilalagay mo rito, 13 drops of blood. Diba? Dito, 12 drops of blood. So, the total number of the drops for each tube should be 25 drops. Yan ba? 25 drops dapat lahat. Kaya kung sa test tube number 24, naka 24 drops ka na ng 0.5% sodium chloride. So, ilang drop ng blood ang ilalagay mo sa test tube number 24? 1 drop. Pag test tube number 23, 2 drops. Sa test tube number 25, may ilalagay ka ba? May ilalagay kang blood? Wala, kasi total number na 25. Naintindihan ba? Okay. Now, so then after putting the blood specimen on each tubes, so you wait for several hours, incubate it, and check kung saang tube nag-start yung hemolysis. Okay? Saan merong hemolysis. So you will also determine the initial hemolysis or partial hemolysis and complete hemolysis. So saan ka mag-start tumingin? From 25 papunta sa test tube number 12. Tignan mo, pag sa 25, nakan mo nilagay na blood dyan. Diba? So, magpapag-control kasi we are expecting that at 0.5, there should be no hemolysis. Diba? Okay, now, check mo, baka may hemolysis na dito sa test tube number 24. Ang initial hemolysis, there will be redness on the fluid, then there is a red cell button, no? Meron pa ring red cell sa ilalim ng ating test tube. Tingnan mo kung saan nagsimula yun, no? So, kunwari sa test tube number 24, clear pa rin, walang hemolysis. 23, wala pa rin. Tinignan mo sa 22, yung initial hemolysis, may hemolysis na ba sa test tube number 22? Ay, wala pa rin, no? Nung sa test tube number 21, nakita mo na, may red na yung, may red na yung fluid, tapos may red cell button. Siya yung unang test tube, kasi pwede mo na makita yung sa test tube number 21, di ba? May red dito, may red cell button. Pwede pareha sila, pero sino nauna? Siyempre, from here, you read here on 25, papunta sa test tube number 12. As if test tube number 21, yung first test tube that you show there is initial hemolysis. So record it. The initial hemolysis happens on test tube number 21. Di ba? Okay, now how, then, tuloy mo pa, hanapin mo naman yung complete hemolysis. So complete hemolysis is the first test tube that shows reddish or redness in the fluid but no more red cell button at the bottom of the tubes. Okay? Nangyari yun sa nakita mo yun sa sa test tube number 16, no? Yung unang test tube. Kasi maaaring makita mo na rin yan sa test tube 15, sa test tube 14, parehas sila. Pero nauna mo nakita yung red na yung fluid tapos wala nang natirang red cell button. Si test tube number 16. Okay? So record it. Test tube number 16, showing the complete hemolysis. Diyan ba? And then after that, so nagkaroon ng initial hemolysis sa test tube number 21, complete hemolysis on test tube number 16. What concentration of salt is in the test tube number 21? So multiply it with a factor 0.02. So the answer here will be 0.42%. Tama ba? 0.42. Then the same thing multiplied by 0.02. This is 0.32%. Okay. Yan. So, ang concentration ng salt na kung saan yung RBC ng pasyente nangyari sa test tube number 21. So, 0.42% yung initial hemolysis, complete hemolysis, 0.32%. So, if this is the result that you obtain, so what is the interpretation? So, our initial hemolysis reference value is between 0.40 to 0.45%, and complete hemolysis may happen at 0.35 to 0.30%. So, in our example, it is normal. So, the red cell of the patient is normal. So, there is no hereditary spherocytosis or there is no spherocytic RBC present in the patient sample. Naintindihan ba? So this is another procedure that we can use, no, in an EOFT studies. Yan yung wala ka pang nakaprepare kasi merong ganun, no, nakaprepare na talaga. May 1% na yun, 0.95, 0.8, etc. So kung 0.5% sodium chloride lang ang available ang reagent mo, so you can do this. Yan ba? Yan. Okay. So that is how we do that is the principle of the EOFT studies. So again, the EOFT is used to diagnose hereditary spherocytosis. Ano ba? Okay, now, sorry. Okay, now, In the EOFT study, one of the part of the procedure is get the hemoglobin concentration or absorbance of the hemoglobin using the spectrophotometrical analysis, no? Using the spectro. Kukunin mo yung fluid, and you will subject that into a cyanide hemoglobin. And then you will get the absorbance. Ganyan, ng bawat test tube, no? Kunin mo yung blood, yung fluid ng test tube 1. Kunin mo yung fluid. Subject it to a cyanide hemoglobin. You know the principle of the cyanide. So you will use a Drakins reagent, mix it with the fluid, no? Around 20 UL of this fluid, and then get the absorbance. You will just get the absorbance. So each test tube, you will do that. You will get the absorbance of each hemoglobin concentration on the fluid. So kung 13 test tubes yan, 13 test tubes din, magsa-cyanide ka. Bawat isang fluid, isa-subject mo sa cyanide. And get the absorbance, and then you will plot it. So in an EOFT studies, we are not only running the patient sample, but we also run a control. Okay? So may control din. Kaya kunwari, ito, no, 13 test tubes yan para sa pasyente. Meron kang control, 13 test tubes din. Then you will also subject the control on an EOFT along with the patient sample. Then you will determine the absorbance of the hemoglobin concentration or content on all of those fluid of the patient as well as the control. Kaya medyo maraming test tubes gagamitin mo. Dahil meron kang isang pasyente lang. No? May mag-run ka ng control. 13 test tube yung ginamit mo sa pasyente. Ito yung kanyang result. No? Tapos 13 test tubes din dun sa control. So each of that will be subjected to cyanide. So meron ka uling 13 test tube para sa cyanide sa patient. Meron ka na naman 13 test tube para sa control. So marami-raming test tubes ginagamit mo rito. You will determine and put it in an EOFT graph the different absorbance of each test tubes. So ito yung result natin. No? We are expecting that the partial hemolysis should happen at 0.40 to 0.45 complete hemolysis. So what I'm saying is one of the protocol in the EOFT is determine the absorbance of all the fluid, absorbance of the hemoglobin content on all of those fluids. Tingnan ba? Then you will put it in an EOFT graph. Yan. Okay, sa EOFT. So ito yung percent hemolysis. Okay? Or minsan nakalagay dito ay absorbance. Okay, percent hemolysis. So you will have a graph. So this B, ito yung control. No? So we are expecting normally, normal red cells should have, hemolysis should be detected, what tube? Doon sa between around 0.40 to 0.45. Diba? Pag 0.5, dapat wala pang hemolysis eh. Kaya, kung nakikita nyo dito, ito yung point where the control will fall. Okay? Ngayon, if the patient is suffering with hereditary spherocytosis, there is an early lysis of the red cell. As you can see here, sa 0.7 pa lang, meron nang na-detect na hemoglobin doon sa kanyang fluid. Dapat wala eh. Dapat mag-i-start yon na meron nang ma-detect na hemoglobin sa fluid between 0.40 to 0.45%. Hindi ba? Eh 0.7 pa lang meron na. Pagdating sa 0.6 meron na. May 10% hemolysis na. Okay? So may absorbance na. 0.20. So kaya itong letter C, this patient is representing that there is an early destruction of the red cells if it is subjected to an EOFT, or there is what we call increased sensitivity to EOFT, or there is an increased fragility and decreased resistance of the RBC. So that's why patient C or the C graph representing that there is spirocytes. So ito, baka meron ngang hereditary spherocytosis yung tao. Or other poikilocyte that may be present in the patient sample that also share the same result with spirocytic RBC, such as macrocytes, macrovalocytes, and elliptocytes. Pwede rin ganyan ang results nila pagka graph na sa EOFT graph. Now, ito namang graph for A is representing RBCs that has decreased fragility but increased resistance to hemolysis. So, sino-sino itong pwede na mga poikilocyte na pwedeng mag-show ng graph A? So, yung mga, if there is a presence of non-lysing RBCs such as target cell, hypochromic RBCs, or sickle cells. Kasi itong mga RBCs na ito, hindi basta-basta nagla-lyse kagad. Naintindihan ba? So, ganyan. Kaya pagka sa EOFT in the laboratory, after we determine what concentration nangyari initial hemolysis at complete hemolysis, hindi natatapos doon. We have to plot it in an EOFT graph by comparing it to our standards or control. Okay? So, yung subject it. Naintindihan ba? So, ang control natin, nag-fall sa graph ng B. Okay? Iwanan ba yun? So that is the principle of the EOFT. So again, EOFT is used to diagnose hereditary spherocytosis. Okay, now there is another test here, which is specific also for hereditary spherocyte, known as the Eosin 5' maleimide binding test. Now, this is a special exam that is used for flow cytometrical analysis. Now, Eosin 5, tawag din nila dito, EMA, no? EMA, it binds specifically on the protein band 3. As I mentioned to you kanina, if there is a defect or abnormality in protein band 3, pwede mag-lead sa hereditary spherocytosis because protein band 3, 4.2, ankyrin, and spectrin, they are fused together within our RBC membrane. So in hereditary spherocyte, pwede maapektuhan yung mga other proteins, no? Protein band 3, 4.2, and ankyrin. So in EMA, specifically, it stains or it binds with the protein band 3 of our membrane. And after applying EMA on our red blood cell, they will be subjected into a flow cytometrical analysis. So loosening or less fluorescence or means that there is a defect on a protein band 3, which may be caused or which leads to hereditary spherocytosis. Okay? Kasi meron ang flow cytometric analysis is dependent also on the fluorescence of a particular protein that you want to measure on the membrane of the red cells. But less fluorescence, so it means there is no number of cells there. And we will be discussing the principle of the flow cytometry. I'm just trying to give you an idea what is Eosin 5' maleimide binding test. This is a type of a reagent that specifically binds on the protein band 3, and then pagka mataas ang fluorescence or yung reflection doon sa, not reflection, doon sa monitor, no? Nakikita kasi sa monitor yon, no? Ng flow cytometry. Malalaman mo kung mataas ang level ng protein band 3 or wala. Pag wala, meron kang hereditary spherocyte. Pag mataas, wala kang hereditary. So hindi hereditary spherocyte. So wala kang problema. So yan yung isa sa ginagamit ng flow cytometry. But we will be discussing the principle of the flow cytometry. Paano malalaman na meron nga ang protein band 3 or walang protein band 3 or kulang yung protein band 3, may deficiency. We'll just discuss. But basta for now, just remember that Eosin 5' maleimide binding test binds with the protein band 3 of our RBC membrane, and then it will be analyzed using the flow cytometry machine, okay, or automated machine. So those are the different tests for hereditary spherocytosis. So spherocytes are seen in the peripheral blood. So pag nakakita ka ng spherocytes, so pwede mo sabihin na baka na may hereditary spherocytosis, or if you want to really confirm, you can use the EOFT studies din. No? So EOFT, kaya lang medyo masalimuot ang pag-prepare ng EOFT test. Okay, now, so there is a question here before we proceed with another special exam. If hepatized blood that will be used for EOFT study cannot be processed within 2 hours. So dapat mag-process within 2 hours. No? Kasi pag hindi ma-process within 2 hours, so there's a possibility that the RBC will not be stable anymore at room temperature. It will cause problem in its appearance or shape, no? Pag sobrang tagal na, ka-expose or nandun sa test tube. Pero kung hindi mo maipaprocess immediately, you can place it on a ref and then it should be processed within how many hours? So your answer will be 6 hours. Okay, so pwede yun. Basta within 6 hours ma-process mo na yung specimen natin. Okay, another review question. Which of the following will have a decreased fragility test? Decreased fragility test. Okay, decrease. RBC stored in EDTA at room temperature for 6 hours. Okay, iron deficiency anemia, thalassemia, sickle cell. Okay, now, ano kaya ang sagot natin dito? Pagka iron deficiency, okay, anemia, thalassemia, sickle cell, lahat sila decreased fragility, increased resistance. Why is it that the iron deficiency anemia and thalassemia and sickle cell have in common? They have all the presence of non-lysing RBC. Like in iron deficiency, there is hypochromic red cells. Hypochromic, if you subject it into an EOFT, as I mentioned to you, will give us a decreased sensitivity or also known as decreased fragility. Okay? Pero increase ang resistance sa osmosis. Yun ang ibig sabihin nun. The same also with thalassemia. What is thalassemia? Microcytic hypochromic anemia yun eh. Hypochromic din. At meron ding mga target cells dyan. So target cells and then hypochromic anemias, they resist lysis. That's why they give us decreased fragility, increased resistance to osmosis. The same also with sickle cell. So that's why all of this will give us a decreased fragility. Okay? So except ito. So the answer will be letter, ano masagot dito? Letter E. Diba? All except A. Lalo na ba, EDTA ang ginamit mo. Ang EDTA, diba dapat heparinized? Ang EDTA kasi, lalo na pagka pinatagal mo yung blood sa EDTA, what will happen to a red blood cell? The red blood cell will swell. Okay? So if it's swollen, it somehow parang naging macrocytic yung RBC mo. So pag nag-swell, sinubject mo sa EOFT, puputok agad. Diba? Kaya increase ang fragility. That is the answer for that question. So those are some of the tests that we use to evaluate RBC membrane abnormality. Okay, so we have PNH. The test for PNH is Ham's test, acidified serum test, sucrose hemolysis, and sugar water test. Then we can also use naman for hereditary spherocytosis, the EOFT study. So those are usually for the RBC problem, no membrane defect. Now, there are also other tests that will now assess the RBC metabolism. We know that the RBC uses two metabolic pathway known as the Ebden-Merhoff pathway, and then we have the pentose phosphate pathway, or the hexose monophosphate pathway. Now, the main enzyme under the EMP pathway, one of the main enzyme there, especially on the third phase of the glucose metabolism, is pyruvate kinase. And the main enzyme, initially on the hexose monophosphate pathway, we still recall that, we have there the glucose 6-phosphate, the hydrogenase. So sa EMP, main enzyme, pyruvate kinase. Sa SMP, the main enzyme is the glucose 6-phosphate. Now, these two enzymes are the most common enzymopathy that we may encounter in the patient's blood. But the most common between the two is the G6PD deficiency. Okay? So may mga tao, kulang yung G6PD. May mga tao, kulang yung pyruvate kinase. So what will be the consequence of that? If you have lacking the synthesis of pyruvate kinase, your red blood cell will be lacking energy, losing the ATP, which is necessary for the normal functioning of our red blood cell. Now, how about if you have a deficiency of your G6PD? So your hemoglobin becomes unstable because your hemoglobin will now be very much prone to oxidization. So yun naman ang consequence pag meron kang G6PD deficiency. So one of the tests that we can use to assess if the patient is suffering with pyruvate kinase or G6PD is known as the fluorescence spot test. Okay, the fluorescence spot test. So that is one of the tests, no? The same also with the autohemolysis test. So with the aid of the fluorescence spot test and autohemolysis, we can differentiate what enzymopathy do the patient has. Is that pyruvate kinase or is that G6PD deficiency? Okay. Now either of the two deficiency, enzyme deficiency, the red blood cell are lysed immediately. So that's why you will have hemolytic anemia. No, kahit na ang dalawa doon, madaling masira yung red blood cells mo. So matetest natin yan with the use of the fluorescence spot test and the autohemolysis test. Now, how about ascorbate cyanide test? The objective of the ascorbate cyanide test is to check the activity specifically of the pentose phosphate pathway or the hexose monophosphate pathway. Because the main objective of the hexose monophosphate pathway is to provide sufficient amount of glutathione, reduced glutathione. Yun ang objective na kung naalala niyo yung discussion natin sa hexose monophosphate pathway, the objective of that is to give our red blood cell enough supply of the reduced glutathione. So may mga enzymes involved din doon, no? Yung G6PD, okay, yung glutathione oxidase, okay? So may mga enzymes na involved doon sa hexose monophosphate pathway. So yun naman yung para sa ascorbate cyanide test para sa pentose phosphate. Okay, so pag-usapan muna natin yung principle of the fluorescence spot test. So from the word itself, the endpoint of what we are trying to determine, the endpoint of this test is fluorescence of the blood. Magpo-fluoresce yung blood tulad dito sa picture natin. So in this particular test, for example, you want to assess if the patient's blood has enough G6PD or pyruvate kinase. So the blood specimen can be a capillary blood. So capillary blood, pwede yun, no? Meron tayong special kind of paper like this. Sa special kind of paper, you will put a drop of blood on those spots. Kaya na spot, no? Okay, maglalagay ka doon, no, ng drop ng blood. Yung sa newborn screening, no, isa sa mga enzyme na dinedetect, kung normal ba yung level ay yung G6PD, no? Pwedeng gamitin yung tinatawag natin fluorescence spot test. So maglalagay ka ng drop ng blood, putting it here on the paper, a special kind of paper. So yan, ang mga drop ng blood dyan. So sa mga spots na yan, so lalagay mo. Okay, now, so for example, you want to check first the presence or the level of the G6PD in the patient's blood. All you have to do is to use a reagent. So naglagay ka na ng blood, no, doon sa paper, sa special kind of paper. Then you will drop a reagent. So merong reagent na ginagamit. Ang content ng reagent na yon ay may G6-phosphate. Okay? So meron siyang G6-phosphate. Ibiyan ba? Yun ang content ng reagent na yun. Okay, now, so if there is an enough level of G6PD in the patient's blood, what will happen here is this reaction. The G6-phosphate plus NADP, okay, and if the G6PD is sufficient or enough on the blood, not deficient, no? magkakaroon ng transformation of the G6-phosphate, magiging 6-phosphogluconate, and the formation of the NADPH. So it will be flooded doon sa drop ng blood, yung NADPH. Diba ba? Now, the NADPH, if it is subjected into a UV light, it will fluoresce. So magpo-fluoresce yan. Diba ba yun? Kaya ang positive result, para masabing hindi ka if deficient ng G6PD, in the fluorescence spot test, dapat yung blood mo mag-glow. May mag-glow siya. So may timing yon, no? Kaya nakalagay rito, time 0, 5 minutes, 10 minutes. So the intensity dapat pa-increase ng pa-increase hanggang 10 minutes. The intensity of the fluorescence of the blood. What causes the fluorescence of the blood? The presence of the NADPH. Now, if the result ay yung intense, i-compare yun sa control, no? Kaya maglaro ka ng test, tsaka positive and negative control. You will compare the intensity of the fluorescence of the drop of blood sa mga controls. So dapat nagko-coincide yung intensity ng fluorescence ng sample ng pasyente doon sa control. Kaya ito yung sa pasyente, ito yung control, at yung negative control. So the negative control should be no fluorescence. So i-compare mo, dapat at 5 minutes, the intensity of the fluorescence almost the same or same with the control, positive control. Pag umabot ng 10 minutes, mas tataas yung fluorescence, i-compare mo rin, no? Pag compare mo parehas halos yung intensity ng fluorescence, then the patient is not suffering with G6PD deficiency. So there is sufficient amount of the G6PD. Hindi ba? Pero pagka yung intensity niya, no, mas intense yung nandoon sa control, positive control, 5 minutes, parang madilim pa, hindi pa masyado nagpo-fluoresce. Pagdating sa 10 minutes, hindi rin masyado nag-fluoresce. So, maaari, meron kang deficiency. Or, if hindi talaga nag-fluoresce, talagang totally you are deficient with G6PD. So, makikita mo naman sa negative control. Kaya, yung intensity at 10 minutes is very important, okay, in reading the level, no? So, pag hindi ganun ka-intense yung fluorescence, so baka kulang or konti yung G6PD mo. Pagka naman talagang hindi nag-fluoresce, so you are really suffering with lacking of the G6PD in your system or in your blood. Okay? So again, for G6PD, the blood should fluoresce. Now, how about for pyruvate kinase? So the same thing, you put the drop of blood on this paper, okay? You will run the positive and negative control along with the test. Then, sa reagent, hindi G6-phosphate ang nakalagay. Ang nakalagay, phosphoenol pyruvate. You will drop phosphoenol pyruvate reagent on the blood drop. And then, if there is pyruvate kinase in the patient's blood, the reaction will happen, no? Yung phosphoenol pyruvate, magiging pyruvate. Yung pyruvate, dahil may lactate dehydrogenase din yung blood, magpo-form naman ng lactate tsaka NAD. Yung NAD ay hindi nagpo-fluoresce. So the lactate and the NAD will be the endpoint of the reaction. NAD will not fluoresce. Ang nagpo-fluoresce yung NADPH. So that's why, in a fluorescence spot test, in order for us to say that the patient's blood has sufficient amount of pyruvate kinase, the blood should not fluoresce naman. Pag nag-fluoresce, ibig sabihin ay, ano nangyari? So pag nag-fluoresce, ibig sabihin, deficient ka ng pyruvate kinase. Diyan ba yun? Kaya sa pyruvate kinase, no fluorescence should be seen. Ano? So baliktad sila. Si G6PD, magpo-fluoresce. Si pyruvate kinase, dapat no fluorescence ang makita sa drop of blood. So pag baliktad naman, si G6PD, walang fluorescence, may G6PD deficiency ka. Pag si pyruvate kinase, no, nag-fluoresce yung blood, okay, so ibig sabihin, you have a deficiency of pyruvate kinase. So that is the principle of the fluorescence spot test. Now, there are certain things that we have to remember whenever we are, meron kasing may mga false negative and somehow, sometimes a false positive result or false normal result, no? Pagdating sa fluorescence spot test. Okay, so for example, this question, which of the following will give a false normal result of fluorescence spot test for G6PD? What is normal? So ibig sabihin, nag-fluoresce yung blood. Again, so ano kaya ang sagot dito? May give false normal result for the G6PD detection. So your answer will be all of that. So if the blood specimen has very high platelet count, high WBC count, high hematocrit count, okay, it will give us a false positive result or false normal result for the G6PD detection. Now, if the hematocrit of the patient is more than 52%, or more than 52%, yung hematocrit, no? There is a quenching effect in the fluorescence spot test. If there is an excessive or excessively high red blood cells, okay, masyadong mataas yung levels of hemoglobin, no? So may quenching effect kasi daw yung hemoglobin doon sa fluorescence spot test. That's why the hematocrit is more than 52%. You have to reduce the volume of blood that you will be using for that particular test. Okay? Now, if naman, mababa naman masyado yung hematocrit, okay? So you have to double naman yung specimen's required volume na gagamitin mo dun sa test. So the hemoglobin has a quenching effect on the G6PD, in the fluorescence spot test, I mean. Pa ba yun? Yeah. So it is very vital that whenever we are reading down yung fluorescence spot test, so we have to be very careful in assessing the time kung ano yung intensity. We compare it around 5 minutes and 10 minutes. Now, if the fluorescence happens beyond 10 minutes, parang biglang nagpo-fluoresce ng konti, no, beyond 10 minutes, the result is negative. Okay? So 10 minutes yung dapat. Okay, 15 minutes na, 20 minutes doon pala medyo nag-fluoresce. Okay, so still that is a negative. That is a G6, kung G6PD on G6PD deficiency ka. Okay yun? Yan. So those are some of the things that may cause false normal, no? Or false normal. Kasi itong mga to, yung mga platelet count, mga WBC count, retic count, meron din kasi ganyan meron din G6PD yan. Baka, kaya ang mangyayari, pag masyadong mataas yung WBC count, kaya platelet count, ang ginagawa, i-spin muna yung blood, tapos tatanggalin yung yung supernatant, yung WBC, the Bafico, before it will be used for fluorescence spot test. Okay, so pwede kasi sa anticoagulated tube, no, for the fluorescence spot test. So that is for detection of difference between pyruvate kinase and G6PD. Okay, now, if the patient is suffering with G6PD deficiency, there is a possibility that there will be the formation of this oxidized form of hemoglobin known as the Heinz bodies. So Heinz bodies can be visualized using the supervital stain such as methylene blue or brilliant crescent blue. But I have mentioned to you that other inclusions of the red blood cells, abnormal inclusions, can also be stained by supervital stain, like the Howard-Jolly bodies, hemoglobin H, and the Pappenheimer bodies. Now, but if you want to really confirm if that inclusion is Heinz bodies, right, you have to use another stain known as another reagent known as acetylphenylhydrazine. So you will be subjecting the red blood cell into this reagent, and then after that, you do a smear and check if there is inclusion bodies formed after you put this reagent on a specimen. Okay, check kung merong inclusion bodies na na-form. Pag meron, Heinz bodies yun. Kaya pang-confirm natin ay yung gamit na acetylphenylhydrazine kung Heinz bodies nga yun, no? Pero hindi, ibig sabihin, pag nakakita ka ng inclusion, positive agad, you have to count the number of the red cells. The inclusions, you have to observe several red cells and check how many inclusions you see on the red cells. So pag positive for acetylphenylhydrazine, so meaning positive for Heinz bodies, you must see that more than 32% of the observed red blood cells showing 5 or more inclusions. Like for example, in one field approximately you're observing around 100 red blood cells, and you see that around 32 red blood cells, or more than 32 number of the red cells there showing inclusions bodies such as this. So sabi mo, positive yon sa Heinz bodies. Pero pagka less than 32%, okay, of the red blood cell, may inclusion nga. Pero less than 32 lang, mga 12 lang, no? Bawat field, nakakita ka lang ng mga 10 o kaya 9 na may mga inclusion nga. Ibig sabihin negative yun for Heinz bodies. So it is not automatically that if you see an inclusion after adding acetylphenylhydrazine, okay, you will report immediately that it is positive for Heinz bodies. So ang pH ng acetylphenylhydrazine nasa 7.6. So it will form inclusions in the red cell if it is a Heinz bodies. Now Heinz bodies is seen in certain enzymopathies such as the popular one is the G6PD deficiency. But Heinz bodies can also be found in other enzyme deficiency that affects the hexose monophosphate pathway, or that is part of the hexose monophosphate pathway. Then it is also seen in certain disorders such as unstable hemoglobin. We see that oxidized form of the hemoglobin. That is now. So Heinz bodies are formed, if you have G6PD deficiency, Heinz bodies are formed after your body is administered by certain drugs. So itong mga drugs na to can cause formation of peroxides that will somehow eventually makes our hemoglobin oxidized. So ito yung mga example ng drugs. Itong Favabins is not a drug, it is a bean, that somehow affects the activity of the G6PD on those susceptible individuals. Lalo pagka deficient yung kanilang G6PD in the body. So yan yung mga isa sa test natin. Pagka meron tayong Heinz bodies, nakikita ang Heinz bodies saan? Usually sa G6PD deficiency. Now, another test for enzymopathy to differentiate between G6PD deficiency and pyruvate kinase deficiency is what we call the autohemolysis test. Now, in autohemolysis test, from the word itself, autohemolysis, if we subject our blood samples on a temperature around 40 to 45 degrees Celsius or 42 to 45 degrees Celsius, okay? Or maybe up to 50 degrees Celsius. But usually, ang ginagamit mga around 45 degrees Celsius. our red cell somehow can still withstand that temperature without lysis and normal red cells. But eventually, it will lyse at a certain point. Okay, pag masyado nang matagal, pag na-exhaust na yung energy or glucose doon sa plasma, malalyse eventually. Pero at 40 to 45, hindi mabilis mag-form yung lysis. Okay? Because a normal red cell have enough energy to sustain the energy. But in certain cases like G6PD deficiency and pyruvate kinase deficiency, pag sinubject yung RBC on that temperature ranges, the RBC immediately lyse. Madali siyang nalalyse. So mag-iisip ka, may early lysis na nangyari at that temperature ranges. So isipin mo, baka G6PD or pyruvate kinase deficiency. Gusto mo malaman, sino dito sa dalawa ang dahilan? Bakit mabilis malyse yung red cell? Nilagay mo sa 45 degrees Celsius, sa 42 degrees Celsius, or maybe up to 50 degrees Celsius. But usually what is the range being used is around the average of around 45 degrees Celsius. Nalalyse kagad yung red cells, madaling malyse, no? So malaman between G6PD ba yun o pyruvate kinase. So you will now get two test tubes. Okay? One of that, lalagyan mo ng glucose. Yung isa lalagyan mo ng ATP. So this, the blood specimen of the patient, lalagyan mo ng glucose yung isang test tube, lalagyan mo naman ng ATP reagent yung isang test tube. Then subject that on the same temperature. For example, 45 degrees Celsius. Subject it there. Okay? 42, 43. So mga ganong ranges, no? Then check for hemolysis. If the hemolysis is corrected, kanina na-hemolyze, no? Mabilis malyse yung red cell. Merong lysis kada dun sa plasma niya. Nakita mo, merong kagad red tinge, no? Madali siyang malyse. Pero nung nilagyan mo ng glucose, na-correct yung hemolysis. Walang hemolysis. Pero still, nung sa nilagay mo yung ATP reagent, hindi na-correct yung hemolysis pa rin. Ang tawag dun sa reaction report na yun ay autohemolysis type 1. So in autohemolysis type 1, glucose corrected the hemolysis while ATP reagent didn't correct the hemolysis. So what is the enzyme deficiency? G6PD or pyruvate kinase? So in this case, the problem is G6PD deficiency. So in G6PD deficiency, glucose corrected the hemolysis, but ATP will not correct the hemolysis. And we call it autohemolysis type 1. If it is reversed in autohemolysis type 2, glucose didn't correct the hemolysis, but the adding of the ATP reagent corrected the hemolysis, then this is seen in pyruvate kinase deficiency. Okay? While if it is no hemolysis, meaning both adding glucose and ATP reagent corrected the hemolytic pattern or hemolysis, okay, after subjecting the specimen at a hotter temperature, okay, na-correct. Tawag ng autohemolysis type 3. So saan nakikita yun? Nakikita yun sa hereditary spherocytosis. That's why G6PD deficiency will show us autohemolysis type 1 result, pyruvate kinase deficiency, autohemolysis type 2 result, and hereditary spherocytosis will show us autohemolysis type 3 result. So with the aid of the use of the autohemolysis, we can screen what enzymopathy might be present in the patient's body or blood. It is pyruvate kinase deficiency or G6PD deficiency. Again, this is a screening test, okay, for G6PD and pyruvate kinase. Running back. Okay, then another test for RBC metabolism is the ascorbate cyanide test. Now, the principle of this test is we will get the specimen, what kind of specimen. Now, what we will do is we add this reagent, sodium cyanide, sodium ascorbate. Now, what is the action of the sodium ascorbate? The action of the sodium ascorbate is that if it binds to the hemoglobin, it lyses the red blood cells, and the hemoglobin will be liberated in the specimen. The sodium ascorbate plus hemoglobin will form hydrogen peroxide. Okay? So what will happen to our blood? The blood will be oxidized. Okay? So it will be oxidized, forming brown color. Magba-brown. Yan ba? So magba-brown. Ngayon, yung hydrogen peroxide, the peroxide formation in the blood will be reduced by the hexose monophosphate pathway. Because the objective of the hexose monophosphate pathway is to provide sufficient amount of glutathione, reduced glutathione, with the presence of glutathione, with the help of the hexose monophosphate pathway, such as glutathione reductase enzyme, as well as, of course, nandun pa yung ibang enzyme like G6PD enzyme. So the sodium ascorbate oxidizes our blood by forming peroxides. These peroxides, if you have sufficient amount of reduced glutathione, because of the aid of the hexose monophosphate pathway, like if you have sufficient amount of glutathione reductase, ay meron kang enough nun, no? So what will happen to your peroxide form in your blood? It will be transformed into non-toxic substance such as water and oxygen. So hindi magubuo yung brown, hindi ba? So there will be no brown color if you have sufficient amount of glutathione, reduced glutathione. So, ibig sabihin, wala kang problema sa hexose monophosphate pathway mo. Kaya ang negative sa ascorbic cyanide test, no brown color. Yan ba? Pag nag-positive, sa positive sa brown, ibig sabihin, wala kang enough reduced glutathione in your blood. Meaning, baka may problema ka sa hexose monophosphate pathway. Maaring may G6PD enzyme deficiency ka or may deficiency ka ng glutathione reductase enzyme. So, ibig sabihin, defective yung hexose monophosphate mo kasi nag-allow ng brown color formation. So, remember, hemoglobin plus sodium ascorbate will form peroxide, and peroxide or hydrogen peroxide will cause brown formation or color in our blood specimen. To prevent the brown color formation, you must have sufficient amount of reduced glutathione so that the peroxides form will transform into a non-toxic substance such as water and oxygen, and it will not lead to brown color formation. Okay? Now, kaya lang, mayroong isa pang enzyme na yung hydrogen peroxide or yung peroxide na na-form after adding sodium ascorbate, meron din isang enzyme na nagtatransform it to form water and oxygen. Now, hydrogen peroxide will also be converted into water and oxygen, but this time, it is not part of the hexose monophosphate pathway, and that is catalase. Okay? So, catalase is not part of the hexose monophosphate pathway, but it helps in reducing hydrogen peroxide to water and oxygen within the red cells. Pero ang tinetest kasi natin dito, yung hexose monophosphate pathway, hindi yung catalase. Kaya para maging specific, para sa hexose monophosphate pathway, yung ascorbic cyanide test, talagyan ng sodium cyanide. What is the action of the sodium cyanide? Sodium cyanide shuts down the catalase activity. So allowing only the hexose monophosphate pathway to do its job to transform hydrogen peroxide to a non-toxic substance such as water and oxygen. That's why, pag nag-form ng brown, positive sa ascorbic cyanide test, ibig sabihin, you don't have enough reduced glutathione, so meaning you have defective hexose monophosphate pathway. So the negative result should be no brown color. So ang ating normal result, no brown. Okay? Ang ating abnormal result, brown formation. Pero pag tinanong sa inyo, what is a positive result of ascorbic cyanide test? Merong brown. Pag ibig sabihin ng brown, abnormal yung HMP. What is a negative result for ascorbic cyanide test? No brown formation. Still red pa rin yung blood. So ibig sabihin, yung hexose monophosphate pathway mo normal. Meron kang sufficient amount of reduced glutathione. So that is one of the tests that we can use to screen the hexose monophosphate pathway. Diba? Okay. Again, those are the key tests for enzymopathy. So we have fluorescence spot test, autohemolysis test, and ascorbic cyanide test.